So here we've done it sort of like the same framework with machine learning and making predictions, but here we did it for drugs. So here the features are features of drugs, and this is the, sort of like this class is about finding drugs, prioritizing drugs, and sort of like drug discovery type of topic. So a lot of the, I guess, the regular approaches for drug discovery is looking for a target, and that target is somewhere in that cell signaling pathway that I mentioned to you before. The problem is that we don't really know whether the small molecule is binding to other things, and the effect is a network effect. So this is sort of like an alternative approach where we try to predict the side effects in this case, but we can also predict indications for drugs based on those features, based on this machine learning approach. So here, this matrix is from a project that is done on the Broad, where they treated these cells with 20,000 small molecules, and then they took images from those cells, and then they visualized the cells using technology called cell painting. And so every image is converted to a set of features that are extracted from the image using some software. So there are 812 features, morphological features. Those are the columns. So the drugs are the rows, and then the features are the columns, and those are numbers for every one of those 800 features. You can also take the small molecule and then break it up into chemical fragments. Did you guys learn about that already in this course? How to do that? So there are libraries that cheminformatics people developed where they take the smile strings of the small molecule or some other representation of the small molecules and break it up to substructures, and then every substructure gets a binary digit, and then it's now become like a binarized vector of 166 flags to tell you whether the parts of the structure exist. And then here it's this L1000 data that I'm going to talk to you about, which is the links data, where the people at the Broad, they use all the same 20,000 small molecules, and they treat them on those cancer cell lines, and they measure how genes are differentially expressed before and after drug treatment. Okay. So those are my features, and what I want to learn is this adverse event reactions. And those are recorded in two databases. One is called CIDR, which is like an effort by a European group that took all of the inserts from drug, sort of like those things that you get when you prescribe a drug. It tells you in there the list of side effects. So they create a dictionary of all of the side effects, and then database. So this is in CIDR. There's also other sources for side effects. The FDA has post-marketing surveillance system. It's called FAIRS, and they keep track on side effects after the drugs are marketed. And so basically you have the side effects are the columns, and then whether the drug induces that side effects is encoded in this matrix. So every row is a drug, and then if the side effect is encoded, is known to be associated with the drug, then you can put a 1 in there. So this is a multi-label classifier. Before the previous slide, we looked at a single vector classifier, that machine learning problem. And then you can learn a function that would take this data and produce this matrix. And the idea is that for FDA-approved drugs, we know the side effects. But for all of those other drugs, we have this information. They haven't been tested in humans yet, so we don't know their side effect profile. So this is important to prioritize what side effect you should look for before you test those drugs in people. And you can also, the same way, you can predict indications. So you can see what those drugs that haven't been tested yet, what things they may work for, what diseases they may work for based on what we know that current drugs are used for. And this is how you evaluate classifiers. So there is a whole lecture can be just done on how you evaluate classifiers. But it's by model distribution of the ability to predict. Shows you that for some drugs this works, and for some drugs it doesn't work. So this is sort of, and there's two groups of drugs that either it works, and this logically can be because some drugs have effects in a cell, or their structure is indicated of what they're going to do to a person, but in some cases the effect is global, and you're not going to be able to get it from just looking at the drug you have to use some type of like a system level of like a whole organism effect of the drug, not just the and the whole thing is noisy, so you have to also be worried about it's not always like a perfect solution. It's a very hard problem to solve.